Imagine a future where reaching 100 years old is no longer considered extraordinary. A future where a person in their 80s could still have the biological characteristics of someone decades younger. Where aging is no longer viewed simply as an unavoidable decline, but as a complex biological process that scientists can study, monitor, and perhaps eventually modify. For most of human history, aging was something we could slow only indirectly. We could treat infections. We could replace damaged organs. We could control blood pressure and cholesterol. We could prevent many diseases that once killed people early in life. But we could never truly interfere with the fundamental machinery of aging itself. Now, that may be beginning to change. Inside every human cell is an extraordinary information system. DNA contains the instructions that help build and maintain our bodies. But those instructions do not operate alone. Thousands of genes interact with one another. Proteins repair damage. Cells communicate. Mitochondria produce energy. The immune system removes threats. And specialized molecular mechanisms constantly attempt to keep the entire system functioning. Yet as we grow older, this balance gradually begins to deteriorate. DNA accumulates damage. Proteins become less efficient. Mitochondria can lose functionality. Inflammation can increase. And some cells enter a state known as cellular senescence. Senescent cells are unusual. They have largely stopped dividing, but they can remain metabolically active. Some release inflammatory molecules and other signals that can influence the surrounding tissue. As these cells accumulate, they may contribute to an environment associated with biological aging. And this leads to a remarkable possibility. What if aging is not controlled by one single biological switch? What if it is a collection of interconnected processes? And if scientists can understand those processes well enough, could some of them eventually be changed? This is where genetic engineering enters the story. One of the most important technologies in modern biology is CRISPR. CRISPR allows scientists to target specific sequences of DNA with remarkable precision. It has already transformed genetic research. But CRISPR is not an anti-aging button. There is no single gene that scientists can simply edit and suddenly make an 80-year-old human biologically 20. Human aging is much more complicated. Thousands of genes and biological pathways influence how our bodies change over time. So the future of longevity is unlikely to come from one miraculous genetic modification. Instead, it may come from a combination of different technologies working together. One area of interest involves a group of proteins called sirtuins. Among them is SIRT1, a gene involved in cellular regulation, metabolism, stress responses, and processes associated with maintaining cellular health. Research in laboratory organisms has made longevity-related pathways, such as SIRT1, scientifically interesting. But there is an important distinction. Something that improves a biological pathway in a cell or extends lifespan in an experimental organism does not automatically translate into dramatically longer life in humans. The human body is an interconnected system. Changing one pathway can affect many others. And sometimes a biological mechanism that appears beneficial in one context can create a completely different problem somewhere else. This is especially important when we talk about cellular survival. A cell that becomes better at resisting damage sounds beneficial. But if that cell has dangerous mutations, allowing it to survive indefinitely could potentially increase cancer risk. And that brings us to another fascinating part of the aging puzzle: telomeres. At the ends of our chromosomes are protective structures called telomeres. They help protect chromosome ends as cells divide. In many types of cells, telomeres become shorter over repeated divisions. When they become critically short, cells can stop dividing or enter a state of dysfunction. So scientists naturally began asking a simple question: could maintaining telomeres help preserve cellular function? One important biological mechanism involved in telomere maintenance is an enzyme called telomerase. Telomerase can extend telomeres in certain cells. But once again, biology gives us a complicated answer. Cancer cells frequently find ways to maintain their telomeres, allowing them to continue dividing far beyond the normal limits of many healthy cells. So simply extending telomeres is not enough. The real challenge would be controlling the entire biological system around them. Scientists would need to know which cells should be repaired, which cells should receive rejuvenating signals, and which damaged cells should be removed. This is why the future of longevity may depend on something even more sophisticated than genetic editing. It may depend on cellular reprogramming. One of the most remarkable discoveries in modern biology was the realization that mature cells can, under certain conditions, be pushed toward a younger developmental state. This work is associated with the discovery of the Yamanaka factors. The implications were extraordinary. A mature cell was not necessarily locked forever into its existing biological state. Researchers began exploring whether some characteristics of cellular aging could potentially be reversed. But complete reprogramming has serious risks. If a cell loses too much of its identity, it may stop functioning normally. Uncontrolled reprogramming can also create dangerous cellular behavior. So scientists have become interested in a much more precise concept: partial reprogramming. Instead of completely resetting a cell, what if you could selectively reset certain aspects of its biological age? Imagine an old computer. Over decades, its system becomes cluttered and inefficient. You don't necessarily want to erase the entire computer. You want to repair corrupted files, restore important systems, and keep the operating system intact. Something similar is being explored at the cellular level. The objective would not necessarily be to turn an old cell into an entirely different cell. It would be to restore some of its youthful functions while preserving its identity. If this could eventually be done safely in humans, the implications would be enormous. But aging is not only about DNA and cellular identity. Our cells also depend on mitochondria. These microscopic structures help produce the energy required for life. As organisms age, mitochondrial function can change. Cells may become less efficient at producing and managing energy. And when many different systems begin deteriorating simultaneously, the effects can eventually appear throughout the body. This is why scientists increasingly view aging as a network problem. DNA repair, cellular senescence, telomere maintenance, mitochondrial function, protein quality, inflammation, metabolism, immune function. All of these systems interact, and that creates a completely different vision of future medicine. Imagine visiting a hospital not because you are already seriously ill, but because your biological systems are being continuously monitored. Doctors analyze your genome. They examine molecular indicators of cellular aging. They measure inflammation. They evaluate organ function. They monitor metabolic changes. And artificial intelligence analyzes enormous amounts of biological data to identify patterns that humans might miss. Instead of waiting for disease to appear, medicine could potentially become increasingly predictive. And instead of treating only the consequences of aging, future therapies might attempt to maintain the biological systems responsible for healthy function. But now we reach the question that captures the imagination. Could all of this eventually allow humans to live for 200 years? The honest answer is, we don't know. There is currently no scientifically established genetic therapy capable of giving humans a 200-year lifespan. The idea of a 200-year human remains speculative. It is a possible future scenario, not a medical prediction. But something important has changed. Scientists now understand aging at a much deeper level than ever before. We can manipulate genes. We can study individual cells. We can observe molecular changes in real time. We can analyze enormous genetic datasets. And we are beginning to develop technologies capable of altering biological systems with increasing precision. If these technologies continue improving over the next several decades, human lifespan could potentially increase. But lifespan and health span are not the same thing. Living to 200 while spending the final century in poor health would not represent the future most scientists are trying to create. The real goal is health span, the number of years a person can remain physically and mentally healthy. Imagine reaching 120 while still being active. Imagine continuing to learn, working, traveling, creating, raising families, and participating in society without experiencing the level of biological decline that we associate with extreme old age today. That would be a much more meaningful revolution than simply adding numbers to a birth certificate. But extending healthy human life would create enormous social consequences. If people lived for 150 or 200 years, careers could change completely. Education might become something people return to repeatedly throughout life. Retirement could move far beyond the traditional concept. A person might have several entirely different careers over a century. Families could contain 6 or 7 living generations at the same time. But longer lives would also create difficult challenges. Population growth, housing, food, energy, healthcare, employment, and perhaps the biggest question of all, access. If advanced longevity treatments were extremely expensive, would they initially be available only to the wealthiest people? Could genetic longevity become another source of inequality? Would humanity create a world where some people could biologically remain young for centuries, while others continued aging normally? These questions may eventually become just as important as the science itself. Because changing human lifespan would not simply change medicine. It could change civilization. And there is another question hiding underneath all of this. If we could dramatically extend human life, would people actually want to live for 200 years? Would a longer life change the way we think about relationships, purpose, career, family, time itself? For thousands of years, humans have lived with the knowledge that life is limited. That limitation has influenced our decisions, our cultures, our philosophies, and our understanding of what it means to be human. Changing the biological limits of life could therefore create consequences that no laboratory experiment can completely predict. And yet, the research continues. Scientists are studying the genes involved in cellular maintenance. They're investigating senescent cells. They are exploring telomere biology. They are developing increasingly sophisticated forms of genetic editing. And they're investigating whether aspects of cellular aging can be reversed. None of these discoveries guarantees immortality. They do not prove that humans will live for 200 years. And they certainly do not mean that aging has already been defeated. But they're changing one fundamental idea. Perhaps aging is not a single unavoidable event. Perhaps it is a collection of biological processes. And if those processes can be understood, measured, and safely modified, then the future of aging could look very different from the past. 200 years may sound impossible today, but science has repeatedly transformed seemingly impossible ideas into engineering challenges. The technology required for a 200-year human does not exist today. The risks remain enormous. The biology is incredibly complex, and there are still more questions than answers. But somewhere inside laboratories around the world, researchers are studying the molecular machinery that determines how our cells survive, repair themselves, and eventually decline. And if humanity eventually learns how to control that machinery, the greatest medical revolution may not be about curing one disease. It may be about changing the biological conditions that allow disease and aging to emerge in the first place. The future may not give us immortality. It may not even give us 200 years, but it could give us something more realistic. More healthy years, more time with the people we love, more time to learn, more time to create, and more time to experience a life that today would have been considered impossibly long. The question is no longer simply, how long can a human live? The deeper question is, how far can we safely rewrite the biology of aging? And if we eventually discover the answer, we may discover that the greatest frontier of human evolution was inside ourselves all along.